lecture covers microbial growth and the prokaryotic cell cycle. The cell cycle is a sequence of events from formation of new cell through the next cell division. And basically it couples DNA replication and partition of the chromosome and actual cell division or cytokinesis. And so that's highlighted in this cartoon here, in which you see a young cell at the early phase of the cell cycle. And this young cell then elongates. And so new cell membrane and new cell wall are laid down. Simultaneously, the chromosomes are dividing. And in the process, the cell reaches some critical length and begins to form a septum. These two pieces of the genome are separated and segregated into the resulting daughter cells so that you end up with two daughter cells of roughly equal size. So this process is referred to as binary fission. Chromosome replication and partitioning has a few key points, and obviously it's very sophisticated and there's a lot more to it, but most prokaryotic chromosomes are circular. There's an origin of replication, and there's a terminus. So again, the origin of replication is where replication of those circular chromosomes begin. There's a terminus where it ends. The replosome is just a bunch of the proteins and things needed to sit on the origin of replication and begin replication. And then other little factors like MREB is an actin homolog that plays a role in determining cell shape, but also chromosome separation. So the question of how do these chromosomes at this point here know to segregate into the different daughter cells. So, and that's facilitated presumably by MREB. And then there's something called FTSZ, which is a tubulin homolog that plays a role in cell division. So FTSZ would form right here in this septum and then allow these two cells to separate from one another. So the process looks something like this when you see the things together. So here's a cell early in the growth phase that's basically starts to elongate. You see the two replication forks at the origin of replication. So here's the origin of replication, and then the replication forks move out in two directions. And so again, that's what you're seeing here. These two replication forks copying the chromosome in two directions. And eventually, again, the cell septates, the two complete chromosomes divide into the daughters, and then again you end up with two daughters with the exactly right amount of DNA. The growth curve for bacteria is pretty straightforward, and it's usually plotted as a log of cell number versus time, and it has 4 distinct phases. So that's what's illustrated in this particular graph. Here on the y-axis on this side is the log viable organisms per mil, and then the x-axis shows you time. This second y-axis shows optical density, which is just a measure of how cloudy a liquid culture, for example, gets. Obviously, as more cells grow, the cloudier the culture gets. Over here, the viable counts, imagine a flask in which you're shaking the flask, for example, and then periodically pulling out samples and measuring the amounts of live bacteria in the sample. And this is something that we would have done in lab, and so I'll give you a lab demo on that. But the general gist of the growth curve is that it falls into these 4 predictable categories. The first category is the lag phase. The second category is exponential phase, and then the third phase, again, is stationary, where the growth doesn't really increase or decrease, and then a death phase where there's a slight decline. And so each of these next slides covers a bit more of the detail of the phases. The lag phase in this particular case is when the cell starts to synthesize new components. Basically what they're doing is getting adapted to the new medium or the conditions that you've put them at. So a lag phase may be long or short depending on how ideal the conditions are or how easy the conditions are to adjust to. And so again, it varies in length for that reason. So again, that's this phase right here, and this could be long and then go up, or it can be really short depending on, like I said, how difficult it is for the microbe to adjust to those new conditions. Exponential phase, or it's also called log phase or logarithmic phase, is the growth in which is the phase in which growth is pretty consistent. Cellular constituents are manufactured at a constant rate. So this is the most ideal phase where the microbe is growing as fast as it possibly can on the nutrients, the temperature, and all the other conditions that are provided for it. It's usually very uniform as long as you don't disturb it. And so in that particular case, the phase is usually pretty smooth as long as the conditions stay consistent. Sometimes you get a growth like this, and then it goes, the curve might go like that and then go up again. And often when we see that kind of that extra little lag, it means something happened during the experiment. Like the power went out and your incubator stopped holding temperature or whatever. So when we see something other than a nice smooth exponential or log phase, that's usually some kind of unbalanced growth. Something happened that changed the situation. There was a change in nutrient content, for example, or a change in some kind of environmental conditions, like the temperature or something, or the amount of shaking in the flask had changed. Stationary phase is where the total number of viable cells remains kind of constant, and typically it's because metabolically active cells stop reproducing. And in this particular case, it's a normal population dynamic. And so again, we're looking at this phase right here where things look pretty consistent. And usually what happens is the cells sort of run out of nutrients. And it's typical of most populations. When they run out of nutrients, they stop reproducing. So some of the possible reasons, again, nutrient limitation, or in the case of aerobic organisms, then limited oxygen availability. It could be the opposite, right? If the organism doesn't like oxygen, it could be the infusion of oxygen that actually stops the growth. So it depends on the circumstance. It depends on the organism. But certainly toxic waste accumulation will happen. So if you grow an organism in a medium that of lactose, for example, the sugar lactose, and the microbe makes a ton of waste products, in this case lactic acid, the microbe can actually poison itself. Or if you're big on fermenting, yeast cells will produce a bunch of ethanol in wine, for example, in the production of wine, and that ethanol can actually poison themselves. So that actually would slow their population down. And certainly when a critical population density is reached, and again, too many physical cells in the presence of a limited amount of nutrients. The starvation responses that we typically see when microbes have reached these phases mainly include morphological changes. And one thing I've already alluded to was endospore formation, in which microbes make these tough, resistant spores, which are typical in really inhospitable environments like the soil. One thing that microbes also do is they may decrease in size or shrink their protoplast, their cytoplasm, or condense their nuclei. And the purpose for this is obviously to create a smaller cell that's not as difficult to feed. The surface area to volume ratio of a smaller cell actually increases. Some cells will actually produce starvation proteins, and by doing that, these starvation proteins do a bunch of things, like protect the DNA or protect the ribosomes, or can glom onto certain proteins or enzymes to maintain their functionality. They go into these long-term survival phases where they again tighten up their cell wall or tighten up their membrane. And one thing that we know that they do as well is increase virulence. So many times when you back a microbe against a wall, it'll get more deadly. It'll produce more virulence factors that can break open more red blood cells, for example, to get more nutrients. Two alternative hypotheses as to what's happening in the death phase are the viable but non-culturable phase, in which the cells are alive but dormant, or a programmed cell death, in which a fraction of the population is just genetically programmed to die. In this particular case, it's kind of like an altruism. You can imagine that if every few cells just pops open and releases their guts into the medium, other microbes have the potential to eat those guts. And so they can actually survive off of the body parts of their of their siblings. Whereas in the viable but non-culturable phase, this is a real tricky one because microbes can still be alive, but they just don't grow on nutrient media. And so in that particular case, this is a really big issue, for example, in the food industry. So if you're a ground beef inspector, for example, and you're sampling tons of ground beef by pure culture isolation, and there may be salmonella in there, but the salmonella could be in this VBNC state. And if you pluck a bunch of the meat and grind it up and plate it, and don't get anything to grow, it's real easy to say, oh, look, the meat's clean. But it's possible that there are these VBNC cells in there. And so what's interesting about the phase is that the microbes might not grow on culture media, but something about the human body can resuscitate these VBNC cells. And so they may actually grow again inside the person and cause them to be sick. So we've started using a variety of molecular tools to detect the DNA or some signature for the microbe rather than looking specifically at live cells. So it's a really interesting physiological state that causes tons of problems, especially from a public health or a food micro perspective.